cardiovascular, respiratory, renal, and digestive systems. Did everybody pick up a set of handouts? There's a clicker, the color, clicker, a handout of today's PowerPoint, the syllabus, of course, and the schedule. Everybody should have those 4 documents. I will ask you to please bring the clicker with you to class each day, just like I have done in the past. Please remember, I'll print out a few more. It's also available on Moodle. If you lose it and you want to print it out on your own, that's fine. It doesn't have to be the color printer, color copy. So, AMP2. I think about half of the class took AMP1 and about half the class did not, which is fine. That's completely fine. We jump off from cell physiology. Those of you who have taken AMP1 know the drill. You know the expectations. And those of you who have not will pick it up pretty quick. So AMP1 focuses on the nervous system and the muscular system, and AMP2 focuses on kind of the rest of the body, right? And so in this class, in this room, we will mostly do physiology, and in the lab, we will mostly do anatomy. Not exclusively, but that's the separation phase. Let me ask you to read the syllabus, please. I know all your professors do say the same thing, right? Read the syllabus and think about how my learning goals, at least how I have expressed the learning goals, and how they align with yours. So let me go through this very briefly. I'll just highlight a few of the highlight a few things. One is that I have set up office hours, so those are times that I plan to be in my office, of course, but those are not the only times that you can meet with me. If that doesn't work, please just send me an email and we'll work something out. I want to be available to you to answer your questions. That's part of what I see as the job. Not just in this room, not just in the lab, not just grading your papers, but working difficulties out with you one-on-one or in small groups or whatever. So please don't hesitate to contact me and make arrangements if you're feeling like you're confused, you're worried, you want to talk about something, you just want a clarification. Did you really mean to say that? This class moves fast. Some of you have already experienced that. So it's important to keep up. It is not successful to think that you can put it off until a few days before the exam and then get it. It just doesn't work. So don't even try is my recommendation. Take one of these seats. Let's see, what else? The textbook is required. There is a copy available at the CST desk if you want to borrow it instead of have your own, but I recommend that you have the required textbook. It's the same as AMP1, so it's works for 2 courses. Same with the anatomy coloring book. I will not be checking your coloring. I won't be seeing how well you stay within the lines. But I think the coloring book is a useful way to learn the anatomy that we're talking about in lab mostly, and also in this room. So the anatomy coloring, it's not a kid's book. It's medical students use this book to relearn and refresh their memory of anatomy. So it's a valuable resource for learning the anatomy, and it will be the go-to textbook that I will go to when I'm thinking about setting up a quiz or an exam question that focuses on anatomy. I will go to the coloring book and say, okay, what should they have learned from the coloring book or from lab, and then design a question based on that. So the coloring book is your source to learn material for the anatomy quizzes and anatomy questions on the exams. So the reading assignments and the coloring assignments are that's your job for learning for the class. 4-credit course, because this is 3 hours a week, and then we have lab. You can see the course grading. So exams will be in class, closed book, you know, typical exams. You've experienced it before, either in CELPIS or AMP1. But doing well requires more than just memorizing. I mean, anatomy is largely memorization. I'll be honest about it. It's learning names. It's learning a language, which is a lot of memorization. Physiology is a lot more than memorization. Physiology means being able to apply a knowledge and understand something so that you could take something that you learned here and apply it here and figure out a problem. So memorization is a start, but it's not enough to do well in this class. You've got to be able to apply what you know to do well. So exams will be in here. I'll probably figure out a way to, maybe I'll figure something out for exams. This room is too crowded for exams, but there's room that we can spread out. I'll figure it out. Quizzes are sort of self-study. So self-timed. You will go, the quizzes will be made available in this is just like AMP1. Quizzes will be made available at the CST desk. You go there, you pick it up, you check it out, you sign it out. You sit down somewhere right there, preferably. Do not take it out of CST, but you can take it to a quieter space if you don't want to work in that commons area. And do the quiz. And then turn it back in at the CST desk. Okay, so that's how you do it. They take about 30 minutes. The quizzes are focused mostly on anatomy, but I could throw in a physiology question there, just like I could throw an anatomy question into an exam. So the quizzes focus on anatomy, and as quizzes and exams approach, I will make sure that you have a set of concepts or information that you need that it will guide your study for that quiz or exam. So the quizzes are self-study. You go, you check it out, you sit down, you take it. There will be a temptation to cheat, right? Because it's anatomy, it's memorization. You'll say, oh, I knew that, and I'll just look it up. Well, that's cheating. And so, and a few years ago, I had an episode of cheating in this class. And it was painful for all of us. And so I will ask you to reduce the temptation by leaving your books and notes and phone somewhere away from you when you're sitting at the desk. Increase the barrier to, nobody wants to cheat, but there's a temptation. I understand the human temptation to do better by looking something up. And that's just not right. And so by leaving your stuff not just at your feet, but somewhere else, the act of getting up and having to walk over, that triggers a, ooh, wait a second, what am I doing? That's not right. And you sit back down and you just do your quiz, okay? So leave your stuff away from you. And I mean, let's face it, this is, it's Earlham. I trust you to do the right thing. But if you see someone with their phone out during a quiz, let me know. I'll follow up. If I see someone with a phone out during a quiz or during an exam especially, I will assume you're cheating. I mean, even if you're texting about lunch, I will assume you're cheating, and we'll talk about it. Problem sets. Unlike quizzes and exams, problem sets are open book, open notes, open colleagues. You know, you can work together on problem sets. You'll turn in your own problem set answers. You can't plagiarize, you can't copy each other's answers, but you can work together. I want you to work together. I think that's part of the learning experience here, is at least for the problem sets, because problems are, they're working on the material and applying it, maybe using some arithmetic, mathematics to answer a question. I want you to work together on it. And so feel free to, you know, form whatever group you want to with the problem sets. Problem sets will be handed out in class, then made available on Moodle, and then turned in at the CST desk on the Sundays that they're due. So quizzes are typically made available on a Thursday and due on a Sunday at like 11:30 p.m. That's a typical schedule for a quiz. Problem sets are made available probably the week before, and then are due at the CST desk at 11:30 p.m. I mean, I know 11:30 p.m. is a weird time to have something due, but that's when CST desk closes for the weekend, and so that's when they're due. If there's a problem, if there's a conflict and you need an extension, please just talk to me. Because the Sunday at 11:30 p.m. deadline is, will I start grading at midnight on Sunday night? No, I won't. So we can be flexible about that. Questions about exams, quizzes, problem sets? Really? Okay. Attendance here is expected. I think you'll learn better if you come to class. Attendance in the lab is required. If you can't come to a lab, you have to let me know ahead of time and we'll work it out. So look at the schedule, please, and look at the rest of your calendar and see if you've got any issues with lab, quizzes, exams, problem set, deadlines, etc. Okay? I would like to make a note that I am being video recorded right now. I'm going to video record every class. This is going to serve two purposes. One is, in the future, when you're not taking the class, I intend to flip some of the classes. How many of you know the expression a flipped course or a flipped class? Okay, not everybody. So in a flipped structure, students watch a lecture as homework and then come to class already having seen the lecture, and then come to class and do problems together. That's so the idea is, instead of coming to class and hearing the lecture, you come to class already having heard the lecture. That's the flip, and then you do the work in class. In a typical classroom, in the traditional classroom, you come to class, we talk about it, and then you go home and do the homework, the work. So that's why it's called flipped. I intend to flip this course somewhat. I don't intend to flip it entirely. But the first step in a flip is having a video lecture. And so I'm after all these years of teaching this class, I'm finally getting around to video recording this class. The other thing that this video recording does, which is helpful to you, is that I will make them available to you online. And so when it comes time to review for a quiz or an exam, you can actually go through the video, and you can go through it at 2 times, you can go through it fast, and catch up the, review. What did Bob say about, mean arterial pressure? And just find that section and review it. Okay? If you miss a lecture, if you miss a class, you can review it on the video. But I still think participating in class in person in 3 dimensions is better than just watching the video. Okay. We talked about academic integrity. The disability accommodations are the same as everywhere else, every other course that you've taken at Earlham. So I'm happy to talk with you. If you have a formal accommodation that you get from Donna Kiesling's office, please, we'll do the thing. That's great, that's fine. Communication, I urge you to communicate with me about anything. And then there is a final note about sexual harassment and assault, which is now standard operating procedure for classes at all. So please read the syllabus and incorporate the information. Please look at the schedule and collate that information against your other deadlines, and let me know if there's any problems. You'll notice on the schedule, those on the schedule are the homework assignments. Either reading, coloring, or a problem set due the Sunday affiliated with that schedule, or a quiz, or an exam. So the schedule is your guide. Okay, so I haven't been using the PowerPoint. So, okay, we talked about the course goals, and let me just click through this. We talked about the books, that you should have them. So we did all of this. So, and this PowerPoint will be available on Moodle after class. All the PowerPoints. So not only will the videos be available, but the PowerPoint deck will be available to you after class. I don't print out the entire deck. I print out selected slides just because, well, paper. Did I problem sets due on Sunday, open book, quizzes, available due Sundays, exams are in class, deadlines. We covered all this. Okay, let's talk about lab for a moment. So half of you have already done an anatomy and physiology lab, and half of you haven't. And you can probably compare notes about it. And those of you who have done it can share your knowledge with the rest of the class. But the point is, the lab is focusing on the human cadaver anatomy. And the main, so some of you have already been inculcated into the brotherhood and sisterhood of anatomists by having worked on a human cadaver. And the rest of you are now joining this, what used to be a brotherhood and is now a community more widely defined. Right? So this is one of the most famous paintings of anatomy there is, the Anatomy Lesson of Dr. Nicholas Pope by Rembrandt. And so people have been doing what you will be doing in lab for 1,000 years. And that makes us part of a long-standing community of anatomists. The goal is to use the body to understand human anatomy, and there's no better way. There is no better way to know anatomy than to have experienced it in 3 dimensions of the human body. So you're going to immerse yourself in the not literally. You're going to engage in with the human body and learn from it. And we will focus mostly on the thorax and the abdomen. That's where the heart, lungs, digestive and kidney, renal systems are. So the other class focuses on the brain, nerves, and the muscles, and this one is mostly chest and abdomen. Because the class is big, we can't even fit really into the anatomy lab. So I'm going to have to split this class is going to get split into half. And so half of you will have lab from 1 to 3, and half of you will have lab from 3 to 5. Some of you have a conflict at 1, and some of you have a conflict at 3. So you have a strong preference for the other section. What we have done in A&P for many years is I run a lottery, a random generator. I will divide this class. The class will get a random assignment, and whoever goes first gets to choose their gets their choice. And so we'll put it up on the board, and so there's 30 people in the class. So each section is going to have 15 students, maybe 16. I'm not going to go past 16. So it's going to be 15-15 or 16-14. It just is. And if you get a bad number, and your only slot is left in the section that you cannot do because of a conflict, we will try to ask for the people who prefer the section they're in, but don't have a conflict, to make an accommodation. And it has always worked. I'm always nervous about it because I don't want to kick somebody out of the class because they can't do the lab section, but I've never had to do it. So I'm hopeful for this class again. So we'll run the lottery, we'll divide the class into half, and so you'll come at 1 o'clock sharp. We will start on time because we're basically squeezing a 3-hour lab into 2 hours for each of them. And so we'll start on time, we'll get to work. In each half of the time, so about approximately 1 hour each week you will spend with the cadaver, and 1 hour each week you will spend doing something else. So for example, in the very first lab, not today's, which we will have. So today we will meet to do the lab sectioning. Next week we will meet and we will start to see the inside of the human chest in the cadaver lab, and we will explore pig hearts for the other half. And so half the lab will start, half of you, well, a quarter of you will do the cadaver lab first, and the pig heart section second, in the second hour of the 2 hours, and then we switch, right? So each week you will spend 1 hour with the cadaver and 1 hour doing something else. It means that not everybody will have exactly the same cadaver experience, because the people who worked in the first hour will do something different than the people who worked in the second hour. But you will have equivalent cadaver experiences, not equal, not the same, but equivalent. So, for example, in the first hour, the people might spend the time cutting the bones of the chest and actually exposing the heart for the first time. And then in the second hour, the people will be removing the pericardium, seeing the actual heart, maybe even taking it out of the chest and holding a human heart in your hands. So those are different experiences, but they are equally powerful. And then the following week, we'll make sure that everybody sees what what groups did after you were in the lab. Questions about lab and this system for divvying it up? So please come to lab today. It's the AMP lab at Stanley 151. It's basically across the hall from the CellPhys lab, and we'll do we'll run the lottery and section everybody in the lab. Those of you who have had AMP1 will be good to go at that point, and those of you who haven't had AMP1, I will introduce you to the cadaver. We won't do work, we will just do an introduction. Okay, so these are the systems we're going to be talking about: cardiovascular, respiratory, renal, and digestive. They are all interrelated, and so I'm going to try to put things together as well as talk about them separately. So human anatomy and physiology stands on the shoulders of the cell physiology that you all have had. And so we're going to have to review some of the most key concepts from cell physiology, but we're going to go at lightning speed. So I'm just going to, lots of things very quickly. It'll be a little bit like drinking from a fire hose, but then you go home and, fill in the gaps that you missed during class. So I understand that this is just, an intense amount of information, but it's required, and I don't want to reteach, and you don't want to reteach CellPhys because you want to get on with the new stuff, the good stuff, with the new stuff, anatomy and physiology. So here are some things that we will be covering in this class that were introduced in CellPhys. And these three that are in bold are the ones that we're going to be doing right now. We're going to get started. And later on, I will ask you to remember things about receptors and second messengers and action potentials and membrane potentials and muscle and contraction. Because in fact, pretty soon we're going to be talking about muscle contraction. That was near the end of CellPhys, but it's very important for understanding cardiac physiology, right? It's a muscle. So this one is coming up pretty soon. And neurotransmission, neurophysiology will come up pretty soon too, because the heart responds to the nervous system. So let's take a brief break now. So I always quick review of membrane transport mechanisms. So this is a slide from cell physiology. We need to talk about simple diffusion of molecules. We'll see why in a moment. And we'll talk about some of the passive and active transport mechanisms that you, hopefully these words are triggering some good memories from cell physiology. So in anatomy and physiology, there are molecules that simply diffuse through the membranes. And this is most important in respiratory physiology because oxygen and carbon dioxide, the gases that are most important in respiratory physiology, diffuse through the cell membranes, from the airway into the blood and from the blood into the airway. So simple diffusion is an important part of the transport systems that we will talk about. Simple diffusion is always downhill, down the concentration gradient. But we're not going to talk about most of these, but like I said, we will talk about oxygen and carbon dioxide. Other important molecules or ions that are transported do not diffuse through membranes, at least not very well, not good enough. And so there are transporters, proteins in the membrane that do the job of transporting those solutes. So let's talk about passive transport. So passive transport, like diffusion, is purely downhill. There's no mechanism for uphill transport. This is down the concentration gradient, from high concentration to low concentration. That's what I mean by downhill. So we have transporters and ion channels that both do passive transport through the membrane. But not through the membrane, through a protein, through the membrane. So channels undergo conformational changes, that you're going to hear this a lot, protein conformational changes to go from a closed state to an open state. So the protein conformational changes are illustrated by the arrow, right? A closed channel with the pores sort of pinched off in some way, and the open channel that allows ions to flow down their concentration gradient. Not always from inside to outside, sometimes from outside to inside. Right? The direction of ion flow depends on the gradient. Remember that from self-is that the membrane is comprised of phospholipids and cholesterol. We probably won't talk about that too much, but occasionally channels are modulated by the lipid environment, and we might mention that. The structure of ion channels is well known. When we get to some important channels, I will not just show this blob cartoon, but actually show some three-dimensional crystal or cryo-electron microscopy images of channels, but I'm not going to do that today. Transporters, passive transporters, like channels and like diffusion, transport ions or other solutes down their concentration gradient. So, for example, a transporter might bind a specific solute, let's say glucose. It's got a binding site for glucose. It's got a pocket in the protein that is especially adapted for forming non-covalent bonds, ionic, hydrophobic, van der Waal interactions, hydrogen bonds with glucose, forming a binding site with some affinity. I'm using words from self-is. Binds it. What happens in this arrow? Protein conformational change. And now the binding site that was accessible on one side of the membrane is now accessible to the other side of the membrane. There may have actually been some change in the binding site that changes its affinity for the solute. In any case, binding and unbinding happens, and when it unbinds, it can diffuse to the other side. So the things that I'm refreshing your memory, trying to get you to think about and remember. Okay, let's unpack this a little bit. So what is involved in this arrow right here? I'll give you a hint. I'm thinking of two things that are embedded in this arrow.
Diffusion.
Diffusion is one. And binding. Because the ion is not bound here and it is bound here. So diffusion and binding. as I already mentioned, binding is non-covalent interactions. So some of the things that you want to review are the non-covalent interactions, bonds. Another concept that you want to review is the concept of affinity. High affinity interactions, low affinity interactions. Happy to answer questions, but I want you to, we went through this in sort of early. I know that was a long time ago for some of you. This arrow suggests that this goes in one direction only. Do you suppose that's true? No. There has to be an arrow in the other direction too. So, passive transport of solutes by transporters, cartooned here. We will talk, this is an important molecule in the brain, which we won't be talking about, in muscle, which we will talk a little bit about when we get to cardiac muscle, but it is very important in digestive physiology. So this is a concept that I'm introducing now, and when we get to the digestive physiology close to the end of the semester, we'll come back to this. So let's do a little bit of something other than me talking. I want you to write down, and I think there's space on your handout or on a piece of paper, what are the key similarities and differences between ion channels and the passive transporters, the facilitated diffusion transporters of ions. Think about what makes them similar and what makes them different. Let's take 2 minutes, and we'll talk about it. And you can work with each other if you want.
I would say the ions like you have a lot of concentration on this side and very little on that side. If the charges on that side is different, it will still go to that side.
You're talking about electrical.
Yes. This step 2 and radius. So the ion is more like balancing. That kind of gradient. Yeah. But the passive transporter is more.
Okay, volunteer. Who wants to mention a similarity or a difference between ion channels and ion transporters, passive ion transporters? Anybody?
They both undergo some sort of conformational change.
Okay, so they have conformational changes. Conformational change exists in both cases.
In both cases, the solute will transfer from our high concentration to low concentration.
Right. So that's another way of saying they both catalyze passive transport. I'm using an enzymology term here, catalysis, because in some ways this transport is like it's an enzyme in the sense that it's lowering the barrier, the energy barrier, for the ion to get from one side to the other. If there weren't the transporter or the channel, the barrier for transport for these ions would be very high because they don't diffuse through the membrane. In the presence of the channel or the transporter, the ions get through more easily. Lower barrier. So from an energetic perspective, it's like an enzyme. Now, it's not changing anything. Well, it is changing something. It's allowing an ion to get from one side of the membrane to the other side of the membrane. That's a difference. That's why I feel allowed to use the word catalysis. So, change in conformation catalyzes transport, let's just say transport from high to low, which means it's passive. Okay, that's another similarity. Anybody else have something? There are other, those are two of the most important, but so there could be something else that you wanted to say, or you could say, would you accept this answer as if this was on a quiz? Would you accept this answer? Now's the time to float those ideas.
They both can still go backwards, though.
Okay, they can both go backwards. That's true. They can go backwards, but the net, so you're absolutely right. The net transport is from high to low. Okay? They can catalyze, like 1 in 1,000, an ion could go uphill, but the net overall transport would be from high to low. That is a good point. Somebody could have said, they're both proteins. I mean, that's sort of embedded in the idea of conformational change. But you could have said, they're both proteins. Would you accept that as an answer? Yeah, that's a true statement. Quite.
How is ions protein?
Pardon?
How are ions protein?
Not ions, but the channels and the transporter are proteins.
Okay. Yeah.
No, ions are not proteins.
Okay.
Well, some proteins are ions, but not all ions are proteins. We're not talking about the ions. We're talking about the transporters, the channels or the transporters being proteins. So, okay. Now, in what ways are they different?
Facilitated diffusion is usually like more specific to a
More
certain-
specific. So, what you're...
the binding site.
So you're saying that transporters are more specific for their solute than channels are for their solute.
No.
No. The binding site for so are there binding sites within the channel? Let's just put that slide back up. I'm not going to erase it, but this cartoon is terrible, because it suggests that the ions are just going through this hole. It's way wrong. The ions are binding to a site on their way, or more than one site on their way through. There are channels that have very high affinity binding sites for their ions. So that is a misconception that this slide from your textbook would allow you to get, which I'm trying to clarify up here. So channels are just as selective for their preferred ion. Well, many channels are just as selective for their preferred ion as many facilitated diffusion transporters.
So would that be something that you could put as a similarity, that they both have like...
Yeah, that's a good, okay, very good. They both have binding sites. That's an important similarity.
So difference though is it's the number of ions can pass through.
Yes, that is the difference. Tell me more.
There will be much more like ions can pass through the ion channels at a given time.
So ion channels transport thousands of ions, let's just say per conformational change. Thousands, but transporters, so this is channels. The facilitated diffusion transporters transport one or a few ions per confirmation. That is a key difference. So when the channel opens, you can erase it out. When the channel undergoes its conformational change, thousands of ions can pass before it goes back to the closed state. With a transporter, binding, conformational change, one ion. Conformational change, binding, one ion. That's the right thing. Okay, so I have some answers here that I put up and that will be available on Moodle, but you got the most important things. Similarities, both passive, ion binding sites, change in conformation. Differences, one or a few versus thousands. So here are the answers in similarities and differences, and here are just some characteristics that are most important to remember about channels and transporters.
So does
Yes.
that mean channels have more binding sites than the carriers do?
Not necessarily. No, they actually, it's that we're getting into the molecular details, and the answer to that is really, I don't want to deceive you into thinking that that's true. It's not true. Okay, so now we're going to transition to active transport. And the most important active transporter is the sodium potassium ATPase. We hit this pretty hard in CellPhys. We hit this pretty hard in AMP1. Let's go through it. So the sodium potassium ATPase is an enzyme in the surface membrane of every single cell in the body. It hydrolyzes ATP and uses that energy to transport sodium out of the cell and potassium in. The detail is that for each ATP molecule hydrolyzed and the conformational changes associated with that ATP hydrolysis, 3 sodiums are pumped out and 2 potassiums are pumped in. That's a detail. So sodium is pumped out, which creates a sodium gradient. High concentration on the outside, low concentration on the inside. Potassium is pumped in, which creates a potassium gradient. High concentration of potassium on the inside, low concentration of potassium on the outside. A potassium gradient. I won't expect you to memorize numbers, but I will expect you to know the directions of the gradients, because everything gets confused if you don't know that. So I do want you to know that sodium on the outside is high and potassium on the outside is low. Sodium on the inside is hot, sodium on the inside is low, and potassium on the inside is high. You need to know that. I expect you to know orders of magnitude, but not the details. So for example, you need to know that the sodium concentration on the outside of the cell is about 100 millimolar, 100 to 200 millimolar. That's an order of magnitude knowledge. It's actually much more specific than that. It's 145 most of the time. And the intracellular is, somewhere between 10 to 15. This is actually on the high side. So I don't want you to don't spend a lot of time memorizing the numbers, but do know the orders of magnitude that we're talking about. So the sodium potassium ATPase, let's just go through it. So what is being illustrated here? What concepts are being illustrated by that arrow? Diffusion and binding, right? What is being illustrated by this arrow? Okay, I'll buy that. Conformational change. What else? phosphorylation, well, okay, sure. There's a better word for what's actually happening here. ATP hydrolysis. ATP hydrolysis. It is hydrolysis. It is, in fact, in the act of hydrolyzing ATP, the transporter becomes phosphorylated. But if you just say phosphorylation, we're thinking of a kinase. This is not a kinase. Okay? Just want to clarify that distinction a little bit. Kinases are enzymes that transfer a phosphate from ATP to another protein. And in this case, the protein is hydrolyzing ATP, and in the act of that hydrolysis, it is becoming phosphorylated. So it's not a kinase, but it is an ATPase. Okay, conformational change, ATP hydrolysis. This up. What is being illustrated in this arrow? Conformational change. And this arrow? Diffusion. And unbinding, or dissociation. Okay, apply your knowledge. Use this so far. Which configuration, conformation, of this protein has the highest affinity for sodium ions? Describe the conformation of the protein that has the highest affinity for sodium ions. Well, I'm asking, so this is now using your knowledge to solve a problem. I'm trying to illustrate things that you ought to be able to do in their exam. Somebody want to volunteer an answer?
Open.
Okay. Kairu?
Go for it. That she asked.
I think it's like the first figure, yeah.
Okay, the first figure. What is it about the first figure that, so the first figure has the highest affinity. So let's describe this first one.
Because inside the cell it's low concentration of sodium, but it's binding to it.
Okay, so that's what tells you that it has a high affinity. The internal concentration of sodium is low, and yet it is binding all three ions. That is true. What's true about the protein? Things like it is inward facing. The sites are facing the inside. That is a description of its conformation. ATP is bound, but not yet hydrolyzed, is another description of its state that is associated with the high affinity binding site. Conformational change, still pretty high affinity. ATP hydrolysis is associated with a change in the binding site that lowers its affinity and puts the protein into a conformation where those sites are now facing the extracellular fluid. So when they dissociate, because the affinity has been decreased by the phosphorylation event and by conformational changes, when they leave to the other side of the membrane. That's critical. Okay, so we now have, and it's trying to illustrate that by sort of changing the diameters of those binding sites. This is the cartoon version of a low affinity sodium binding site. You see that? It's a cartoon version of it. What are these guys? Those are the potassium binding sites. So phosphorylation of the protein lowers the affinity for sodium and increases the affinity for potassium. It's now facing the outside where the potassium concentration is low, but it has a high affinity for potassium, so what happened, so we're going, let's see, let me draw the arrow. It goes from here to here. What happened in that arrow? Sort of hidden. Trick question. Diffusion and binding, right, of potassium. Potassium is bound. It binds from the outside. So, well, there it is, right there. Sorry, it's right there. This is potassium diffusing and binding. Okay, so let me clear that. Potassium diffuses and binds to the high affinity potassium binding site. What is going on here? Phosphate unbinding dephosphorylation is what we call that. So when potassium binds, it changes the affinity. There's a conformational change that is kind of hidden, but it changes the affinity of the phosphate binding site. And the enzyme dephosphorylates itself. Dephosphorylation. It's not really just unbinding. It's actually, there's a covalent bond that's broken. Dephosphorylation. So potassium binding triggers dephosphorylation, which now what's embedded in this arrow. Potassium binding, dephosphorylation, conformational change, changes the affinity of the potassium binding site, lowers its affinity, changes the conformation of the protein so that it's now facing the intracellular solution, so that when potassium unbinds, it unbinds to the intracellular solution, and changes the affinity of the sodium binding sites. Well, that associated with the ATP binding changes the affinity of the sodium binding sites, and now we start the cycle all over again. So these are most of the mechanisms. We've just gone through molecular mechanisms of the sodium-potassium ATPase. What does it do for us? It creates the sodium gradient, it creates the potassium gradient. We're going to need to know, we need to use that for many things. Questions about this?
Does having the ATP bound versus just having the phosphate bound effect, have a difference?
What do you think?
Probably.
Yeah. But ATP bound and ATP having phosphorylated the protein makes a big difference.
Okay. So each change is another
Yeah, it
change
is.
in affinity?
So some of these conformational changes are not, so we're taking snapshots of something that is more of a continuum, right? So you can say, well, wait a second, this conformation looks almost identical to this, but this one has ATP bound and this is the phosphorylated state. But if you look closely, you can see there's a difference in conformation. So it's a cartoon, but it is trying to illustrate the answer to your question. We can't only talk about sodium potassium ATPase. We need to also talk about at least one other, well, two others actually. The first one is the calcium ATPase. So I could ask you a question, what is similar and what is different about the calcium ATPase, about calcium ATPases, plural, we're going to talk about two at least, and the sodium potassium ATPase, the sodium pump. Well, let's just, some of you may remember, you can even just use the names to answer this question. What's the similarity between the two of them?
Both of them use ATP.
They're both proteins. I heard that somewhere. What's another similarity? They're both ATPases. They both bind and hydrolyze ATP. What's another similarity?
Pump against the
Conformational change. Is that what you were going to say? I jumped on too soon. Yeah, they both do conformational changes. What's the difference?
One is important, the other is the antipode.
What is what?
One is important, the other is the antipode.
Let's not use the words uniport and antiport yet. neither of them are uniporters or antiporters. Sodium potassium ATPase does transport sodium and potassium, but it's not a antiporter
If they are
Be-
hydrolyzed.
because it hydrolyzes ATP. The calcium transporter, the calcium pump, the calcium ATPase, is not a uniporter because it hydrolyzes ATP. But what you're getting at, what Mia's getting at is the idea that the calcium ATPase is transporting calcium in one direction, and the sodium potassium ATPase is transporting sodium in one direction and potassium in the other direction. Good idea, but the use of the language would cause me to make some corrections. Okay, so calcium transport. One transports sodium and potassium, that's the sodium pump, and the other transports calcium. So I'm going to draw up a real simple cardiac muscle cell. Can't get more simple than that. Obviously, that's not the way it works. And I'm going to draw another membrane compartment like that. And so, I'll give it a nucleus. There's some DNA. Who cares about DNA? What's the name of this membrane? Let's just use the language to make sure we're talking the same language. The plasma membrane or the surface membrane? I will accept that for both. That's the plasma membrane or the surface membrane. What's the name? So this is a nuclear membrane, so it's actually a nuclear. It's got two membranes with some pores in it. Okay, just be a little careful about it. It's got chromatin on the inside. It's got nucleolus. Whatever. What's the name of this membrane? Does anyone know what I'm trying to cartoon? Remember, this is a muscle cell, heart muscle cell. Jasmine.
Is it the sarcoplasmic reticulum?
It is. So I'd say it with gusto. It is the sarcoplasmic reticulum. Where are calcium ions in this picture?
It's in the circle.
Where is calcium high and low? High inside what?
The sarcoplasm.
It's high, it's high inside the SR. Where is it next highest in concentration?
Outside the
In the extracellular space. And where is it the lowest? In the cytosol. What are some numbers here? The calcium concentration on the outside is 1 to 2 millimolar. The calcium concentration in the SR is 10 to 20 millimolar. And the calcium concentration in the cytosol is 0.1 micromolar. So 0.0001 millimolar. Right? So a big gradient. We got a big calcium gradient. The numbers are small numbers of millimolar, but the gradient here is huge and the gradient here is huge. What made those gradients? Pumps. Calcium ATPases. So we're going to talk about two types of calcium ATPases. We're going to put a pump here. It is a calcium ATPase. So what goes on either side of those two arrows? ATP gets hydrolyzed ADP. It transports what? Calcium. Which way do I put the arrow? Does the arrow head go on the top or the bottom of that line? Top, because it pumps calcium out of the cytosol. And that is called the plasma membrane calcium ATPase. What's that? Right, let me You have a loop. Now what is it? It's a calcium pump. So just like the other one, hydrolyzes ATP to ADP, uses the energy to pump calcium. Which way is the calcium being pumped? From the left to the right or from the right to the left? You're from which way? That way or this way? That way. Calcium is being pumped into the lumen of the SR, creating the calcium gradient. This is called the sarcoplasmic endoplasmic reticulum calcium ATPase. So now, yes,
Can you
John.
repeat that?
The name?
Yeah.
Sarcoplasmic endoplasmic reticulum calcium ATPase. These two proteins are similar in the sense that they're both calcium ATPases, but they're not identical. Why don't we just call this the sarcoplasmic reticulum calcium ATPase? It's because it also exists in a very similar form. It exists on the endoplasmic reticulum. So we lump the name together into SERCA. And I will accept SERCA as a correct answer to what's the pump that pumps calcium into the lumen of the SR? I don't think I would ever ask you that question. That's too simple. So, all right, so calcium transport is important, and we're going to be hitting this hard when we get to cardiac muscle real soon. There are proton gradients that we're not going to talk about too much, so I'm going to go very quickly. So we're going to not focus on proton gradients, but there are also proton gradients. But here, let's just do some cell phys. What concept from cell phys is embedded in these blobs? Let's just, what are those blobs trying to signify? What do you think of when you see those blobs? Hmm?
The ring and
Subunits.
Couldn't hear.
Protein domains or something?
Domains, subunits, those are the words I'm thinking about, right? These are subunits. What's the name of the type of protein structure that is important when we talk about protein subunits? Just trying to remember some of the language from cell phys.
Is it tertiary?
Tertiary and quaternary structures. Right? Layering on some cell phys. That was the short answer to that simple question. So now, I think the last of the content, and then we'll do some clickers, is to talk about secondary active transport. Now we are going to get to symport and antiport. There are proteins, and we're gonna need to have there's going to be an important one in the cardiac muscle cell coming up real soon, that do active transport. So when I say they do active transport, what must happen for a protein to catalyze active transport?
they go against the
They push something against its gradient. They pump something uphill. They transport something from a low concentration to a high concentration. That's active transport. When I say it is secondary, what do you and it's illustrated on this cartoon, what do you know it is? What's the difference between secondary and primary active transport? Active transport does the same thing, right? It pumps something uphill. It creates a gradient. This is also capable of creating a gradient by pumping something from low concentration to high concentration. What's different about those two things?
What powers?
What powers it?
Yeah, what powers it? What powers primary? ATP hydrolysis. What powers secondary? Another gradient. Takes the gradient that was created by primary active transport and uses it to pump something else up its concentration gradient. So here we have an example of a sodium glucose co-transporter. We're gonna need to know this one when we get to the digestive system. Binding sites. So let's go through this. Diffusion, binding, conformational change, dissociation, unbinding. A sodium binding site, a glucose binding site, coupled conformational changes. The protein only undergoes the conformational change when both sites are occupied. It's got a sensor. It knows. It's a protein. It doesn't know anything, but it can. Its conformation and its activity depends on the presence or absence of the solutes in their respective binding sites. So it only undergoes the conformational change from outward facing to inward facing when both sites are occupied. The sodium concentration on the outside is high because the sodium potassium ATPase made it so. The concentration on the inside is low because the sodium potassium ATPase created that gradient. So the sodium gradient is being used to pump glucose from high concentration outside, say after a meal in your digestive system, to low concentration, say inside your intestinal cells. And why would it be low in your intestinal cells? Well, your intestinal cells is hydrolyzing all that glucose to make the ATP that was necessary to make the sodium potassium gradient and to do all this transport. So glucose is hydrolyzed. I mean, metabolism happens. We'll talk about that some, but self is metabolism. Glucose is metabolized, so the concentration is low. Glucose was just digested from your meal, so the concentration in the outside of the cell is high. And so glucose is being transported, well, in this case, both, so this is a funny example. Glucose is being transported down its concentration too, but this is secondary active transport in this example. So I sort of screwed this up. Concentration of glucose isn't always high in the intestine. So right now, before you eat lunch, the glucose concentration outside of the cells in your intestine is low. You haven't eaten. And yet this transporter can still pump the concentration from low concentration of glucose to a higher concentration inside the cell because it does secondary active transport. It uses the sodium gradient to pump glucose. The sodium gradient came from ATPase. So let's do some clickers. Try to use some of these concepts. Oh, I wanted to say, so there's another important one. There is a sodium-calcium exchanger in the and it's got a name, the sodium-calcium exchanger in the surface membrane of your heart cells. It uses the calcium gradient, no, it uses the sodium gradient. It uses the sodium gradient to pump calcium out. So it assists the plasma membrane calcium ATPase. You see how that is an assistant to this plasma membrane calcium ATPase. This is an antiporter, and this is a symporter. Because in this case, the secondary active transport is in the same direction as the thing that is being transported against its gradient is in the same direction as the thing that is being transported down the gradient. And in this case, the thing that is being transported up its gradient is in the opposite direction from the thing that is pumping. down its gradient.
So will this only work in situations where the gradient of the one that's powering it is stronger
Yeah.
than the other one, or?
Yeah, it will. It would
Okay.
only work if the energy of the gradient of the thing that is providing the energy is greater than the energy of the thing that is being transported. Yeah, sure.
Say the name of that again.
The name, this is the sodium calcium exchanger. Okay, so let's do some clickers. Which of these transport mechanisms require protein conformational changes for normal function? Okay, good. I see a lot of fully correct answers. They're all correct. They all have protein conformational changes. Which of the following, directly or indirectly, provides energy for maintaining ion gradients in human cells? Directly or indirectly? I think the best answer is all of them. ATP, that's direct, primary active transport. Fats, sugars, and proteins are hydrolyzed to make ATP. That's how you make ATP, is you digest fats, sugars, and protein to make ATP. Respiration, cellular and whole body digestion and respiration. NADH, what's that? That's the high energy electron carrier, one of the high energy electron carriers in the mitochondrial electron transfer chain. This is a self-is trick question, not trick, completely true. NADH contributes its high energy electrons, builds the the proton gradient across the inner membrane of the mitochondria, which is used by the ATP synthase to make ATP. So that contributes to ATP synthesis. Why is this true? Well, plants get their energy from photons. Photons are created in the sun. So we all are, in fact, we all depend on solar energy at a certain level. Even though we can't photosynthesize anything, we eat plants, or we eat animals that eat plants. Okay, so this is a little tricky, but that's the correct answer. Let's do this and then we'll call it a day. 2 minutes. In what ways are primary active transport and secondary active transport alike? So, similarities and differences. We're going to make this one 1 minute, right? Anybody? Similarity or difference? Call it out. Evelyn. They are powered differently. So yes, that's true. It's a true statement, but it's not a complete statement. So tell me a little bit more.
Secondary is powered by what previously established gradients. Second one, and primary is powered by.
That's right. Secondary active transport uses a gradient to power it. Primary uses ATP. hydrolysis to power. That,
Okay, but could you also just say like as a similarity, they both require some sort of energy in order to?
sure, that's a similarity. They both require some energy input. That's got to be true. They both what? So they both transport something. Something is transported uphill. I'll just use uphill as a shorthand for from low concentration to high concentration. Jasmine.
Then the secondary, it goes like there's usually two like molecules or speeds that go with it, or in primary there's just like one required.
Except the sodium potassium ATPase really requires two, both sodium and potassium, in order to complete the cycle. So I could come up. So I see where you're going. It's not as accurate as these things, but it's not wrong. It's just not quite as complete.
They both use proteins that undergo some sort of conformation.
Proteins, conformational change. We could be more specific. They're both membrane proteins, right? Yeah. So the obvious ones are still true. So let's see what I wrote. I was trying to answer my own question. Similarity, unlike move solute up the concentration gradient. That's the gradient. Use energy. Conformational changes. Different. ATP versus gradient. So, see you at 1 o'clock. This PowerPoint, the full deck will be available on Moodle by then.